Hello, everyone. It's a pleasure to be speaking again at this year's Bionano Symposium 2024. I am going to be speaking about the framework for the clinical implementation of optical genome mapping in hematologic malignancies. My name is Dr. Adam Smith, and I'm an assistant professor in the Department of Laboratory Medicine and Pathobiology at the University of Toronto. Firstly, I would just like to recognize the fact that this work is the work of an international consortium of multiple institutions across Canada, the United States, Spain, Belgium, and the Netherlands and Finland. And this group has worked very hard to really try to put all of the ideas that we have about the clinical implementation of optical genome mapping in the context of hematologic malignancies into a framework. And I'm very pleased to tell you as well, I know that probably some of you have heard me speak about the framework in other talks, but in fact, this framework is now published. It was just published recently, January 2nd, 2024. And this is the paper. It's published in the American Journal of Hematology. And down in the bottom right corner, I have noted the PMID for those of you that are excited to go out and look it up right away. Why did we want to create a framework? Well, we know that official guidelines can take years to be developed based on other similar implementations for laboratory technology like microarray and NGS. And we felt like that our experience as early adopters could help other labs implement more quickly and take advantage of collective international experience. And the other thing that happens a lot is when we develop these new technologies, we get bespoke implementations in different laboratories. So everybody's doing the same technology, but they're doing it all a little bit differently, or we're not all using sort of the same thresholds and cutoffs. And so what we really wanted to try to do is in three major sections in terms of validation, quality control, and analysis interpretation, provide some guidelines for people that they could use so that we could get some more harmonization very early on in terms of OGM adaptation. So we can use this sort of roadmap, hopefully, to take a lot of the questions and things that we struggled with early on and help people adopt the technology. So we've detailed the three sections that we want to talk about, including that of validation, quality control, and analysis. And I'm not going to belabor the discussion on validation because we do talk about it in quite some detail in the paper. But we try to break it down into 5 sections that most of you that are familiar with doing validations will be familiar with the kinds of steps that need to be taken. And we've outlined in more detail in the paper some of the really critical points and some of the approaches in terms of, there are some challenges in, say, how do you compare certain types of karyotype data, for example, to OGM data. And so we've outlined some strategies in which you can do that. And so in each of the sections, whether it be the assessment of clinical utility, the proof of principle study, method validation, clinical validation, or implementation, there's lots of really good information that you can use to help you in your implementation of OGM. In the quality metrics section, we've tried using a lot of tables that you can see here and in the paper as well. We've tried to give you all the key quality metrics that we feel are important as a consortium of OGM users that have done a lot of OGM, and how we use those numbers to help us make decisions about whether or not we can safely report data. So we use a lot of the we use the molecule quality report, which is in the analytical phase. So after data collection from the flow cell, and then we look at the targets from the molecule quality report to assess whether or not we feel the data is good enough quality to move on. Then from the post-analytics side, once the assembly, usually in this case for hematologic malignancies, it would be the rare variant assembly. Once that's being performed, then we're looking at these post-analytic quality metrics. And then ultimately that takes us to an OGM clinical report. And your clinical report maybe looks a little bit different depending on how you want to design your report or how your software works. However, a lot of these reports based on accreditation organizations and the criteria that are required, such as CAP and their checklist items, you're going to have several items of information that you need to have on this report, including a results summary, your ISCN nomenclature, and some kind of interpretation of the result, et cetera, and some quality information about the result. And so as you can see here, the ISCN, I just wanted to really quickly point out as a side note that the genome mapping nomenclature paper is also recently published, and you can get that here. The PMID is down here. And so one of the nice things here is, of course, OGM is going to be new to our clinical community. And so giving them good information about what is happening with this sample, what the quality metrics are, whether they pass or not. So again, we have this template where you can see the quality metric and you can see sort of the guideline number in brackets to the right of it. And so you can go through very quickly and you can say, you know what, look at all of these numbers. These are all great. These are all within the expected parameters that we would want to see to have a quality analysis. And so we can look at all of those things. We can also look at making sure that the result and the sex of the specimen that's submitted, so the sample and the analysis match the indication, of course, taking into account any issues that you might run into. For example, one of the things that happens a lot in hematologic malignancies is post-transplant situations where you get sex mismatch transplants. So you can always take that into account when you're looking at these quality metrics. And so finally, to come to our general approach to analysis, what I wanted to do is sort of lay that out in the broad steps, give a very quick example of how we do that, and then talk about how we've actually applied it to myeloid malignancies. And so this is our general approach to analysis, and it's basically 3 steps. So we want to assess for large cytogenetic abnormalities. And these are things we're talking about: interchromosomal rearrangements, intrachromosomal rearrangements, including inversions and homolog translocations, and copy number abnormalities greater than 5 megabases, aneuploidies, etc. So this is stuff that's going to jump out usually on the circus plot for you right away. It's going to be fairly easy to see. From that point, and this is also most of the stuff that's going to be included in any kind of assessment of genome complexity. From there, we're going to look at copy number abnormalities between 500 kb to 5 megabases. So this is 500 kb is the limit that's set by default by the copy number caller in the software, the copy number algorithm, up to sort of the limit of what we consider for the large cytogenetic abnormalities. So this is the sort of intermediate region. And there are some interesting biological problems here, of course, because we're looking for a lot of cytogenetic abnormalities that we're very comfortable with seeing on a karyotype, such as del5Qs, del7Qs, del20Qs. And then the question that comes up is, well, what happens when they're not cytogenetically visible, but they're actually still a deletion in that region? So you still got a del5Q, but it's actually not visible by karyotype. And then, so how small is a del5Q? How small can it be to still have the same prognostic significance? And so these are, with any high resolution analysis, these are questions that the field is going to have to answer. From that step, we then move on to small SVs. And so these are the small dot SVs that are in the circus plot. And there's usually a fair number of these, and we want to be able to assess them quickly to see if there's anything that's relevant. And what we've been using is a region file. So we have a myeloid region file that we apply, and then that really brings genes that are important in myeloid disease biology up to the front, so we can see those and we can evaluate them quickly. Just to give you a couple of examples of things that we have from the paper when we go forward, when we start looking at analyzing samples. What we want to be able to look for is when we're looking for example, a translocation. You can see, again, the 2 reference chromosomes here. You can see the hybrid map in the middle. You've got lots of labels on both sides and good alignment. When we see things like this, for example, this makes us much more nervous. So you see you've got a large variable region in the middle here that looks like there's probably some kind of repetitive element. Plus, then you've got less than 10 labels on the right-hand side. You've got some yellow bars here indicating that there isn't really good alignment with some labels. So there's a mismatch in some of those alignments. So this is the kind of alignment that makes us very, very suspicious. So we've again detailed in the paper exactly how to sort of go through this approach and look at these different alignments to make sure that you're confident in the calls that you're making. And then, of course, when you're looking at these rearrangements, you can also show all of the hybrid maps. So you can, in fact, see both of the reciprocals. If you think you have a reciprocal translocations, it's always good to make sure you can see them both. So you can see the map for the derivative 9 and the map for the derivative 22 against the reference to chromosome 9 in the case of a translocation 922, for example. And then, of course, if you don't have a balanced translocation, but an unbalanced one, again, what would that look like? And so again, you can see things like you can see copy number gain at the breakpoint of this translocation between chromosomes 4 and 6, and copy number loss on the chromosome 6. And then again, when you look at this translocation here, you can again see the copy number and the copy number plots in the genome view. So again, we've detailed all of this stuff in the framework paper to help give people an idea of exactly how we've approached analysis and how we move through it. We've also provided a table which really sets out what we think are the best filter settings, and we've given some advice about why it is we think those are the best filter settings, and when we think it's okay to actually loosen them up a little bit. And so one of those examples is actually when you're looking for deletions and they're at the threshold of what the software is able to call based on the confidence limits at the recommended setting. So what happens then, for example, you can see this very clear deflection in the copy number data here, and you can in fact see that there's an SV call between the 2 points from the start of the deletion to the end of the deletion, which isn't called using the recommended filter confidence. However, we can loosen the recommended filter confidence by setting it to all, and then you'll see the call being made here very clearly. And so this is a situation where the lower limit of detection for this SV call is just slightly better than the copy number algorithm. There's these slight differences, but these are things that as you run more samples and you get to see more data, you'll become more comfortable with. And so, of course, we also have proposed the tiering of variants as was originally proposed for microarray analysis in the McHale et al. 2019 paper. And so we've made some slight adaptations, of course, for OGM, including having to what exactly it is that we're going to be counting in terms of getting up to assessing genome complexity. So we have guidelines for tier 1, tier 2, tier 3, and tier 4 abnormalities. And you can see here that when we look at genome complexity, we're really interested in variants in these categories, whether they be known abnormalities that are in a particular disease classification, or whether they're just somatic changes that occurred. Because when we karyotype, we don't actually make that distinction. We just count up the number of abnormalities no matter what they are. So we're doing the same thing for OGM. We're going to count everything from tier 1 to tier 3A, which these are bigger than 5 megabases. And then these ones here that are smaller, of course, we're not going to count those because we wouldn't have counted them before by karyotyping. So this allows us to keep our assessment of genome complexity consistent with previous technologies like karyotyping, and doesn't require, and while it may be true that really looking at how we assess genome complexity is another question that's important, we do need to be mindful that we do need to collect a lot more data about that and try to figure out the best way forward in terms of dealing with a high resolution assessment of what genome complexity means. So that's a future question, to be sure. So when we apply the framework, for example, here's some data. You can see our circus plot. You can see an SV call here in the middle ring with an accompanying copy number call. So this is a deletion on chromosome 11p. So we're just looking for large cytogenetic abnormalities in our first step. And we have a little look around, and that's basically all we can see. So there we go. We've got our first abnormality. The next step is we move into assess copy number abnormalities from 500 kb to 5 megabases, of which we don't find any. So that's fine. So we're moving on to our third and final step. And you can see here just quickly as you look at this ring around the outside with all the SVs that are called, what we're going to do from this step to the next step is we're going to apply a filter using a bed file with myeloid genes that we believe are relevant to the analysis in this particular case. And then we're going to apply that filter. And what you're going to see is that a lot of those dots disappear. And we're left with 4 basically that we have to analyze. And so there's a couple here that really don't have any good evidence that they have any impact. So we're going to discard those right away. We've got a very interesting RUNX1, looks like a RUNX1 duplication in the body of the RUNX1 gene. We don't really have a lot of evidence about these. So again, this is tier 3. We really don't know what they mean. But of course, here we have a KMT2A PTD. So we know what that means. And of course, we're able to annotate that on the report as well. And we've done some work with our filtering with bed files. We have both a pan-cancer file and a bed file that's myeloid specific. And when we use a comparison on 70 AML cases using both of those analysis approaches, basically by using the myeloid file instead of the pan-cancer gene list, so 140 genes roughly versus about 1,100 genes, what we see is that basically we're just not going to evaluate a bunch of tier 3 and tier 4 findings that with the myeloid region file that we would have had to evaluate with the pan-cancer file. So we're not really seeing any tier 1 or tier 2 abnormalities that are being eliminated as a result of going down only to the myeloid file. The one exception here in the tier 2 data was one case with a BRCA1 deletion that was eliminated because BRCA2 is not in our myeloid region file. And it's a kind of a tricky point. Again, another thing that we're going to need to discuss probably a lot as a community is how much we want to report potentially germline abnormalities that may have other genetic counseling implications. So that's to be discussed in the future talk. And so I also just wanted to very quickly talk about our AML clinical validation, because in our AML clinical validation, we have applied all of the principles of the framework in order to do this validation. So we performed this validation. We had about 70 patients in the initial validation. We found 150 abnormalities using standard of care techniques and 186 with OGM that met the reporting criteria in the framework paper. Therefore, our overall diagnostic utility for OGM compared to standard of care, we see about a 10% increase, which is great. But we also see a lot of cases where OGM detected a tier 1 and tier 2 biomarker that was missed by standard of care, and this was in 36% of cases. And so I'd like to show you this data in the form of an oncoplot. And basically, this is these are the cases across the top. This is the category of the karyotype as it was originally classified. And then these are the cytogenetic and molecular abnormalities as they are currently classified in the 2022 European Leukemia Net classification. And so what I really just want to highlight, because I don't have a lot of time to get into all of the data here, but what I really want to highlight are these blue squares that you can see on the oncoplot. What they represent is cases where OGM has made a significant finding that was missed by standard of care. So for example, there were a lot of cryptic rearrangements that we didn't appreciate originally because we weren't able to see them using the standard of care technique. So we saw some cryptic KMT2A rearrangements, MECOM rearrangements, and NUC98 rearrangements as a highlight. We also saw SVs that overlapped genes that are on the molecular list, which usually are loss of function. And so, for example, the first one there is a B-core deletion. And so whether you mutate B-core or you delete it, you still get the same loss of function. So this is an area where I think that, again, a lot of work needs to be done in looking at the prognostic impact of these particular abnormalities. So looking at the reclassification rate between the 2017 and the 2022 ELN, we've done that as well, 25% reclassified. And this is actually pretty consistent with other papers that have looked at reclassification rates between 2017 and 2022 without doing any additional studies, just looking at the reclassification rate using the different guidelines. And also 22% of the cases had OGM findings that were significant in terms of them actually leading to reclassification as well. So our reclassification rate is a little bit higher than that in other validation studies of these two guidelines, just because, again, we're finding more information that was found in the standard of care. And so, finally, we also have a couple of things where we've compared the percentages of the findings in our cohort of the types of abnormalities we had compared to published literature estimates. And we actually find that comparatively, our numbers are pretty close. The exceptions here are these abnormalities where we're seeing a lot of cryptic changes, like cryptic KMT2A, cryptic MECOMs. And we're seeing the numbers a little bit higher than in the published literature. And this may be the case that we're actually really seeing more stuff because we're actually looking at higher resolution. Oh, and of course, there are emerging biomarkers like the KMT2A PTD, which you can see here, which we've detected in samples, which is very challenging to detect the PTD. There's a lot of interesting components to it. It's fairly straightforward to do by OGM. Other methods have some challenges, and we have a paper coming out soon that's going to highlight a lot of that as well. And so I just wanted to finalize on this point about these are the validations that I was talking about the ELN 2022 and 2017. There's a few that have come out. There's one from the American group at Oregon Health and Science University, and another European group on the right-hand side. So if you're interested at all in looking at those are those papers that you can have a look at. And I really wanted to take this opportunity to thank my development team at the University Health Network, especially Olivia King, Anna Baptista, and Shabnam Salehi Rad. And I really just wanted to thank everybody for the opportunity to speak today. These are my acknowledgements, and I look forward to more questions and interesting discussion. Thank you very much.